Yeah, so thank you very much for inviting me to present at this meeting. So the main question addressed in our study is how to identify dependencies in genome-wide functional screens. And as we all know, the genome-wide functional screens offer the opportunity to identify druggable candidate targets. And this opportunity is especially valuable for those tumors which are driven by oncogenic events represented by transcription factors. Transcription factors are notoriously hard to target. And as an example, I can give you the MECAN, for example, MECAN amplified neuroblastoma, or Ewing sarcoma, which is driven by EWS fly. So the recently discovered CRISPR Cas9 functional screen strategy provides a very powerful tool for the identification of high throughput dependencies across large compendia of tumor lineages. And the advantage of using such type of screening is that the rate of off-target effects is very low. Now, the genes which are differentially, in these CRISPR Cas9 studies, for example, or in general in the functional screening procedures, the genes which are differentially expressed between, the genes which have differential dependencies between lineages are considered to be essential for those lineages. Unfortunately, there are some significant computational challenges for identifying this type of genes which are essential for lineages. And one of the major challenges is that the dependencies actually can be shared among tumors, amongst subsets of tumors from different lineages. And another challenge is that dependencies are, in general, described not by only one functional mechanism, but by a combinatorial essentiality effect of genes which are involved in multiple mechanisms. And in general, the methods which are based on linear analysis, like Pearson correlation, are not always able to capture nonlinear relationships, and they are not always suitable for working on these types of data. So just looking to these standard procedures like the differential marker analysis between lineages, or pathway enrichment, which gene set enrichment analysis, which differentiate between lineages, or even correlation network analysis. They are estimated, and sometimes we show that they are unable to actually extract the meaningful information, reliable information about dependencies. So what we are doing instead of looking to this standard procedure, we are applying deconvolution strategies for identifying better coordinates which describe the biological dependency data. And as you know very well, the basic scheme of such decomposition strategies to start with the data, which is described by features and samples, and in an unsupervised way, identify reliable directions in the data, which are described by the features, and then project the actual data, the original data, onto these directions. And then, if the directions are more reliable for interpreting the data, then do the mining within this projection. So, two basic methods for deconvolution are perhaps well known among the computational biologists. The first one, principal component analysis, is the most used method for data deconvolution. And as you know, it projects the data onto orthogonal components of highest variation. So the variation is estimated as second order correlation. So this method is meant to be able to extract linear dependencies from the data, Gaussian dependencies in the data. And as you can see here, for example, if the data has this structure, we have here a 2D data, then principal component analysis would find first this direction of highest variation, and the second one is orthogonal. But these directions are not correlated with the actual trends in the data. And the other method, basic method for data deconvolution is independent component analysis. It was developed in 1994 by Common, and it is meant to solve the blind source separation problem. So it is meant to unmix the data into a linear combination of independent factors. Now, independence based on central limit theorem is estimated as non-Gaussianity. So this method is able to identify the factors which are nonlinear and independent based on nonlinearity. And as you see here in this toy example, it is able to find correctly the trends in the data. Now, as a principle of proof how these deconvolution methods work, let's look to how they perform on well-known data. It's a CD8 T-cell lineage in the differentiation map of hematopoiesis database, which is also known as TMAP. The data was published back in 2011 in Cell by Aviv Regev's group. So this data has about 20 samples, umbilical stem cells, in different stages of differentiation for CD8 T cells. And we have 4 categories, naive and central memory, and 2 subcategories of effector memory. And here we show how PCA performs on deconvolving this data. So you see PCA here. These are the top 3 components which separate best the data, but you see the lineages are not recovered. While independent component analysis, here we see, again, 3 components which are best for separating the samples in the data, is able to separate the lineages. And you see here we have the naives, and then we have the central memory, and then we have the other 2 branches which correspond to the effector memory. So we are confident that PCA is able to recapitulate the linear relationships in the data, while ICA is most suitable for identifying the nonlinear relationships and, in principle, the intricate, complex relationships for lineage unmixing. So the major study I wanted to discuss about is about using the CRISPR Cas9 screening for revealing the new dependency in MECAN-amplified neuroblastoma. And the study was performed within the collaboration of many groups from Dana-Farber Cancer Institute, from Harvard Med School, from National Cancer Institute, and from University of California in San Francisco, and is currently in press at Journal of Clinical Investigations. So why we chose neuroblastoma for identifying dependencies? We chosen because neuroblastoma is one of the most aggressive tumors in children, and it is the most common extracranial solid tumor in childhood. There are about 600 to 700 new cases per year. It occurs in young children. The median age of diagnosis is 22 months. The cell of origin for this disease is known. It's a neural crest cell. And you can see in the image here how neural cell, tumoral cells look. They have some rosettes inside. Unfortunately, the disease is diagnosed in most of the cases when in high stage. And high stage is very difficult to treat the treatment is not only invasive, it is very painful. It involves a lot of procedures, starting with chemo, surgery, radiation, transplant, bone marrow transplant, immunotherapy, and differentiation therapy. And the prognosis is very poor. So for this disease, in the current time, high stage has a prognosis around 50% for 5 years. Now, there are not that many drivers associated with neuroblastoma, but there is a specific subtype of neuroblastoma called MECAN amplification neuroblastoma, which is identified in 30% of the tumors, and the prognosis is very poor. So we conducted a study on the CRISPR Cas9 screen on Havana Library. This data is publicly available. It was very recently published in October 2017 by the Broad Institute. And this data describes 26 lineages of 300 cell lines, out of which neuroblastoma is represented by 11 cell lines. And for this data, we employed the independent component analysis. We applied a method which is called fast ICA, and we identified top 100 independent components. And this method is well known in the literature. It was developed in 2000 by Hyvarinen in Finland, and it is based on finding in an iterative gradient descent approach the independent components estimated through non-Gaussianity via negentropy. And we added also a consensus scheme out of this algorithm in order to make it more robust. So here we show the results of applying this method on our CRISPR Cas9 screening. We identified 3 top components, independent components, which are able to better discriminate between neuroblastoma cell lines, highlighted in red, and all other cell lines. And a common denominator of all these 3 components was that they are enriched in a module which is associated with the PRC2 polycomb complex. So that was very encouraging, because amongst the genes which are in this polycomb complex, EZH2, the major gene, is druggable. So the 3 genes of the polycomb complex, the main genes, are found as leading edge genes in this independent component. And in contrast, when we applied to the same data a linear decomposition approach by PCA, we couldn't recapitulate the findings, and in fact, neuroblastoma could not be well separated by the others. And PRC2 was not among the principal components which are separating the cases from all the others. And we then validated the results through a semi-supervised approach by performing single sample GSEA on the samples in the CRISPR Cas9 study based on quorum protein database, and we found that neuroblastoma is indeed on top. It scores as top cancer category with respect to enrichment in EED EZH2 complex. And the group, Stegmaier lab group, which is the main collaborator for the study, did a very comprehensive experimental validation. Here we show just a few glimpses in which support the EZH2 neuroblastoma dependency in vivo and in vitro in both genetic and chemical approaches with the guides, SG guides, and we also shown that in mice, the tumor volume shrinks after the tumors are treated with an EZH2 inhibitor. In this case, we use GSK126, and we also experimented with several others, like JQ1. Okay, so in conclusion, we highlight here that ICA and, in general, nonlinear deconvolution methods are able to are effective exploratory methods for the identification of novel druggable dependencies in CRISPR Cas9 genome scale screenings. And we are currently expanding these applications to other disease subsets, and we also expanding to other screening strategies, and we are also looking to single-cell data. And the major collaborators for this study, which have done all the experimental work, which was massive, come from Stegmaier Lab at Dana-Farber Cancer Institute. Well, thank you very much for your attention, and I'm happy to take some questions.